Hello, my dear students. I hope you all have been doing well. Today we are going to start first part of our course 713 that is about introduction of medicinal chemistry. In this part we are going to start our first topic, that is receptor theory. So in this lecture 1, part 1, we will study about receptor theory. So, as the name of this topic is indicating that this topic is about receptor, where we have to study about uh, different theories of receptor. But dear student, before starting this receptor theory, or before starting understanding about receptor theories, we should first have to know about what are receptors and how receptors work, and what are ligands, and what are the chemical nature of these receptor and ligands, and where these receptors are usually found. And we also have to understand about different classes of receptors, and how these classes have been defined or subdivided, and what are the structure of receptors. And we should also have to know about the chemical basis of the receptor, uh, chemical basis of binding of ligand with the receptor. So let's start from here. So what are receptors? First question is, what are receptors? Actually, receptors are the protein molecules, or maybe sometimes they are glycoprotein, that are either embedded in the plasma membrane, or they may be found in the cytoplasm, or sometimes they are also present in the nucleus. And what do they do? The receptors actually transfer or mediate exogenous signals from the outside, that is from the outside, and any chemical exogenous signal in the form that is mm, initiated in the form of a ligand from the outside of the cell surface to the inside of the cell surface till the nucleus. Why they means why receptor mediate these signals from outside to inside? Because this is the signal is important for cell to perform its work. We will see it later, how the signal transfer and how receptor work, and why signal is important for a cell. And here is a simplified picture of the receptor that is embedded in the plasma membrane. And it is showing here that a ligand that is in the form of epinephrine is binding with a receptor, and subsequently this signal that is in the form of a ligand, or a message that is in the form of a ligand, is received by a receptor, and then subsequently this signal transfer inside the plasma membrane in the form of different biochemical changes, and ultimately this will go to the nucleus, and this will be end on cell specific responses. So now we will see what are superfamilies of receptors. Receptors are classified into different superfamilies on different bases, and first base is their function. So on the basis of function of receptors, receptors are classified into four superfamilies, and all the member of these superfamilies are having journal structure and journal mechanism of action, but some individual member of each family having some variation on the basis of amino acid residues or amino acid sequence or the size of their extra and intracellular domain. We will also see it in next slides. What is the extracellular and intracellular domain of receptor? Just we have to focus right now here that what are the uh, how many superfamilies are receptor having and how on what basis they are classified into different families. So as I told you, I'm repeating for you again here that receptors uh, are the protein molecule that are protein molecule or sometimes they are glycoprotein in nature that are find to embedded in the plasma membrane, or they may be present in cytoplasm, or they may be present in the nucleus. And what do they do? They transfer an exogenous signals in the form of a ligand that is initiated in the form of a ligand from outside the cell to the inside the cell for cell to perform its work in form of specific cellular responses. And the ligand is a molecule, a chemical, a molecule or a chemical that when bind to the receptor, it enables receptor to mm, transfer the signal that is ligand is carrying from outside the cell to inside the cell. We will also uh, uh, study about classes of ligands and what are ligands in my next lectures. So the other basis of classification of receptors are on the basis of endogenous ligand. So on this basis, receptors are classified into two types. One is cholinergic receptor and other is adrenergic receptor. So all those receptors that bind with acetylcholine, it is a neurotransmitter, it is a signal, and it is a ligand. So all those receptors that bind to the neurotransmitter acetylcholine are termed as cholinergic receptor type. However, all those receptors that bind to the neurotransmitter adrenaline or noradrenaline, uh, they are termed as adrenergic type. So, on the basis of the endogenous ligand with which the receptor bind to initiate a biochemical changes or to initiate a signal from outside the cell to the inside the cell, um, the receptor are classified on two types. One is cholinergic type and other is adrenergic type. Cholinergic type receptor are all those receptors that bind with the neurotransmitter acetylcholine, and this neurotransmitter where it is found in our nervous system, in our brain. So the other type of receptor that bind with endogenous neurotransmitter, that is adrenaline or non-adrenaline. These receptors come as adrenergic type receptors. Okay, so first class is on the basis of first type of classification of receptors are on the basis of the function they perform. Other type is on the basis of endogenous ligand they bind, and the third one is according to the type of genetic code and exogenous ligand. So dear student, I wish to explain you here the ligand. As I told you that we will study about ligand in my next lecture. But I wish to explain you here somewhat about ligand. Ligand are the chemical molecule. They are chemical in nature. They may be protein in nature, and they may be endogenous or they may be exogenous. You have to know. You have to just consider it. Ligand is a molecule that when bind with a receptor, it transfer the exogenous signal via receptor to the inside of the cell. So these neurotransmitter are the endogenous ligand that I have already explained you about the acetylcholine neurotransmitter and adrenaline or noradrenaline neurotransmitter. These neurotransmitter are found in our brain, and the ligand may be exogenous means we can means consume these ligand by our food. We can take means the drugs we take it, this is one of the form of, you can say, ligand, because it drug also bind with the receptor and initiate its response. So on the basis of the exogenous ligand, um, the receptor are divided as nicotinic cholinergic receptors. These nicotinic cholinergic receptor are those all receptor that bind with exogenous nicotine. So where nicotine is present, you all know that cigarette is also containing nicotine. So it is a form of a ligand, and this nicotine bind with nicotinic cholinergic receptor. So on the basis of the exogenous ligand, the receptor is subdivided as nicotinic cholinergic receptor, or receptor may be bind with muscarine. On this basis, we will say it call it the muscarinic cholinergic receptor. The muscarinic cholinergic receptor further divided into five main types, and on the basis of the endogenous ligand, if this endogenous ligand acetylcholine will bind with the cholinergic receptor, we term this receptor as acetylcholine cholinergic receptor. So now we will see what are these, how many superfamilies of receptor are existing. So receptors are having four superfamilies, and superfamily one is consisting on all those receptors that are fast acting receptor. These receptors are also termed as ligand-gated ion channel or ionotropic receptor, because when uh, any ligand uh, for bind with the receptor, they initiate the transfer of ion, for example sodium chloride, potassium. So they transfer this ion from inside cell to outside the cell, or from outside the cell to inside the cell. So on these and this is very fast means action of the cell, and it happens within a milliseconds. So due to this means speed of action, these receptors are um, termed as fast neurotransmitter or ligand-gated neurotransmitter, because they only open when uh, any ligand bind with the receptor. And these are termed as ionotropic receptor, because these receptor when open their channels, so they initiate the transfer of the ions. As I told you earlier, then the superfamily 2 is the slow transmitter, or we can say we also say them as hormone receptor or G protein coupled receptor. So why we call it G protein coupled receptor? Because G protein is a type of a protein that is linked with this receptor. So we will one by one in my next slides we will be seeing about description of each type. But here you just have to know that the second type or second superfamily is based on all those receptors that are having slow speed, and they are um, hormone receptor, or they are linked with an enzyme or protein. We call this enzyme as G protein coupled receptor, G protein. So on the basis of this G protein, we name these receptor as G protein coupled receptor. Now, third superfamily is all those receptors that are growth factors, or these like the insulin. We say these family as insulin and growth factor receptors, or these all receptors means either it will be growth factor receptor or it will be insulin receptor. They link with an enzyme that we say as kinase. So on the basis of the kinase that link with this receptor, we term these receptor as kinase link receptor. So why we are calling these as growth factor receptor? Because when these receptor are present normally on the cell surface, I'm giving you example here like PDGF receptor, platelet derived growth factor receptor, or epidermal growth factor receptor. So these receptor or they may be insulin receptor. All these receptor, what they do, they are mostly present on the cell surface. When they bind with the ligand, for example, platelet like growth factor receptor bind with the ligand platelet uh, PDGF, and creatine drive growth factor, and the EGF receptor, epidermal growth factor receptor, will bind with epidermal growth factors. So when these receptors bind with their cognate ligand, so what they do, they initiate biochemical changes or cellular changes that will ultimately result on the cell growth, so and proliferation. So on these basis we term these receptor as growth factor receptor. And you know as the insulin receptor, when these receptor bind with the neurotransmitter insulin, so what happened, the insulin transporter that are present inside the cell that transport outside the cell, and they what these uh, transporter do, they take up glucose from outside the cell to the inside the cell, and then inside the cell this glucose utilized to create the energy inside the cell. So I have explained you why we say them growth factor receptor, why we say them insulin receptor, and why we say them kinase link receptor. Now fourth family is steroid receptor like thyroid receptor. They may be vitamin in nature, and fourth family we also include nuclear receptor. So as they, why we are saying is this thyroid or steroid, because these receptors are hormone in nature. They may be vitamin like vitamin D receptor, and the nuclear receptor because these receptor uh, reside in the nucleus. So most usually there's a hormone receptor and vitamin receptor reside in the nucleus, and these hormone receptor and vitamin receptor, these bind with their ligand that will be, for example, for hormone receptor that will be hormone, ligand will be hormone, and the for vitamin receptor the ligand will be vitamin. So now one by one we will be means understand and will study about the structure of each superfamily of receptor and the general structure of each superfamily of receptor, how they means what are the composition of these receptors and how they work. We are starting from superfamily 1, superfamily type 1. So this superfamily type 1 or also called as fast neurotransmitter receptor ligand-gated ion channel or ionotropic receptor. First we will understand about their structure, and then we will understand about their function. So we are here taking an example of nicotinic acetylcholine receptor, and this receptor is composed of alpha subunits, two alpha subunits, and the rest beta subunit and gamma subunit. This receptor, nicotinic acetylcholine receptor, are composed of five subunits. This receptor find to be embedded in the plasma membrane, and the subunit are arranged themselves in a way so they can form a channel uh, uh, by from which the ion can easily pass from outside the membrane to the inside of plasma membrane. And second most important thing that is connected with these receptor are their both N and C terminal reside in the extracellular space. We will see it later why these two terminals are very much important for a receptor for their activity. So mm, first thing to understand or consider it here, that the receptor that are belonging to the superfamily 1 are fast acting receptors, as their name is suggesting here. They are ligand gated ion channel, means they are having a channel like in this receptor nicotinic acetylcholine receptor. Their subunit arrange themselves in a way that they are making a channel that is spanning throughout the plasma membrane for the entry of the uh, nutrients, or we can say that for the entry of the ion from outside to the inside the cell. And they are ionotropic because of this. They are transfer. They work to transfer their means ion from outside and inside the cell. And these are these receptor are consist of four or five subunits, as you can see here, or sometime they are each subunit is made up of 20 to 25 amino acid residues. So this is about the structure mm, of the superfamily type 1, and how they work when the ligand, for example, in this case, here is the ligand. What is the ligand here? Acetylcholine. When the ligand bind with the alpha subunit, this is we have to remember that this the all these receptors that are ionotropic receptor or ligand-gated ion channel or fast-acting neurotransmitter receptor, the ligand must have to bind with two subunit of the receptor. So you can see here the acetylcholine is binding with two alpha subunit. When what happened when the ligand bind with the alpha subunit of this receptor, uh, it due to this binding, the receptor get the channel get open, and then sodium and potassium ion transfer from outside the cell to the inside the cell and produce their effect. What effect they produce? If these acetylcholine receptor are present in the brain, so what happened? The acetylcholine and neurotransmitter, in that we say it a ligand because it is a chemical messenger and this and a chemical signal, and this signal is have to means perform its work. So when this acetylcholine bind with this receptor, so sodium goes inside, and this enable neuron to be depolarized, and fire and action potential. And if these receptors present in endothelium membrane, so what will happen? Or they may present in at neuromuscular junction. So what they will do, they when the acetylcholine will bind with the alpha subunit of this receptor, what will happen? The channel will open, and then calcium will go inside, and this calcium will bind with calcium calmodulin protein, and then this will then um, uh, this will activate with a nitric oxide synthase enzyme to synthesize nitric oxide. So this is about this will be the about of their mechanism. We will now we will go to understand about the superfamily two type of receptor, that are G protein coupled receptor, hormone receptor, or neurotransmitter receptor. We term these receptor as G protein coupled receptor because these receptors are uh, to perform their work. The receptor coupled with a protein that we term as G protein, and these receptors work as somewhat slow as the ligand-gated ion channel. So we are from as a slow transmitter receptor, and here we are taking the example of a muscarinic acetylcholine receptor. First, we will understand what is the structure of these receptor, and then we will understand how they do their work. So these receptors are composed of a single polypeptide chain that is about 400 to 500 amino acid residue. This single polypeptide traverses across the plasma membranes uh, multiple time and make the 7 subunits, each subunit composed of 20 to 25 amino acid, and this 7 subunit group together and remain embedded in the plasma membrane. And the ligand binding site of this receptor lie between this means group. Uh, so now we will see how these receptors work. So these receptors may be work to stimulate an action, or they may be inhibit the action. So due to this, these receptors are divided into two. One is the stimulatory receptor, or one is the inhibitory receptor. So the stimulation and inhibition of work of these receptors based on this G protein, that is remain binded inside the plasma membrane in inactive form, that is GDB bound form, and it is present away from the receptor during inactive state. So what happens when a ligand bind to the receptor binding site? So it causes the conformational change in the receptor. So due to this conformational change, what happens? This conformational change attract this G protein. So the G protein that is present with the GDP bound form, that is inactive form in the cytoplasm, then this exchange to the GTP bounded form, that is the active form. And what happened when due to the conformational change, the receptor attract alpha subunit of the G protein, and due to the binding of GTP with this active form of G protein, due to this GTP, this alpha GTP subunit detach from other beta and gamma subunit of this G protein, and goes to activate uh, two enzymes. One is PLC and one is AC. PLC is phospholipase C, and AC is adenylyl cyclase. So because of this activation of the enzyme, means either G protein activate PLC, or either it will activate adenylyl cyclase. So based on this activity of G protein, these receptors are divided into as stimulatory or inhibitory, because when the G protein activate PLC, PLC convert PIP2 into PIP3, and this PIP3 goes into the intracellular calcium stores and release calcium. Uh, and this calcium is involved in muscle contraction. We will be seeing the destruction of each uh, action of this receptor, or the action of this G protein in next slides. So here we can see that when the ligand bind with G protein coupled receptor, what happens? It activates a G protein. When G protein activate, it activates phospholipase T. Phospholipase T. What this phospholipase T do? This phospholipase T uh, activate PIP, and this and mm, break this PIP into PIP2 or diacylglycerol. Diacylglycerol remain bind to the plasma membrane, however, PIP2 convert into PIP3, and this PIP3 is actually a second messenger, and it will go to the intracellular calcium stores, and then what it do, it bind with the um, uh, it goes to the endoplasmic reticulum that is having intracellular calcium stores. So this endoplasmic reticulum is also having the channel, that is chloride channel, calcium channel. So these second messenger goes to the calcium channel, bind with the calcium channel, and then enable these calcium channel to be open. When these calcium channel will be open, what will happen? The calcium will release in the cytoplasm, and then the calcium will produce its cellular response. And what is the cellular response of calcium in the skeletal muscles? It will enable muscles to be contract. So this is the one function of the G protein. I told you previously in my previous slide that G protein may activate uh, phospholipase C or adenylyl cyclase. So here we saw that how this G protein activate phospholipase C, and via activation of this receptor. After the binding of the ligand to the receptor, how G protein activate phospholipase C. Now in the next slide we will see how after binding of ligand with the G protein coupled receptor, how this G protein will activate the adenylyl cyclase. So when the ligand bind with the receptor, what happen? Alpha subunit of G protein will be activate, and then this subunit will activate enzyme adenylyl cyclase. This adenylyl cyclase will make the cyclic AMP from ATP, and this cyclic AMP is a second messenger that will activate protein kinase A. This is an enzyme, and this enzyme activate various downstream signals. These signals goes to the nucleus, where these signals enable various protein to synthesize, and that protein would be means involved in cell proliferation, cell migration, and cell division. So the another work of this G protein coupled receptor is that this G protein coupled receptor, when activate after binding the ligand with the receptor, what happened? They activate the alpha subunit of the G protein. This alpha subunit goes to the channel, nearby channel, and then this alpha subunit of G protein enable this channel to open. So what happened, that ion goes inside the cell, and these ion, as already told you, for example calcium, sodium, these are involved in various cellular or cell biomolecular uh, processes. The third superfamily of receptors are growth factor receptors, or we say them like kinase linked receptor or insulin receptor is one of the example of the superfamily 3 receptor. So these receptors are composed of an alpha subunit that is bound to the beta subunit. This beta subunit having extracellular and intracellular domain. The extracellular domain of this beta subunit having variable amino acid that is based on the type of the ligand that will be the bind with this um, N-terminal region of this receptor. However, the intracellular domain of this beta subunit having tyrosine kinase activity. What does tyrosine kinase activity means? The intracellular domain of this beta subunit of this kinase linked receptor having multiple tyrosine residues. So what do this tyrosine do? When receptor bind with the ligand, when receptor bind with the ligand, then these receptor get dimerized. This is the one receptor and this is the other receptor, means other uh, receptor, and when then uh, this is the one monomer and of receptor, this is the other monomer of receptor. When ligand bind with this receptor, then receptor get dimerized. And what happened here, these multiple tyrosine residues that are present in the intracellular domain of this beta subunit of the receptor, this tyrosine residue activate trans-activate each other, means the tyrosine residues that are present in this monomer of the receptor, it will activate this side of the receptor, and the tyrosine residues that are present in this monomeric region of the receptor will activate this this site of tyrosine residues. So that is the mechanism of their activation, and what will be the result of this activation. uh, uh, we are taking an example of the insulin receptor. When the insulin receptor bind with this when insulin bind with the insulin receptor, so what happened then, this receptor activate various enzyme, downstream enzyme, and when these downstream enzyme activated, then they, what they mm, activate this intracellular glucose transporter, and these intracellular glucose transporter uh, transport towards the cell membrane of the um, cell, and then what happened, when these transporter comes on the cell membrane, then glucose bind with these transporter and then transport inside the cell, and then glucose is involved in various energy requiring processes, energy metabolism, and provide energy to the cell. So this is how these receptor work, and this is the structure of the type 3 receptor. We will study about the superfamily type 4 receptor. These are nuclear receptor, and these receptor are also termed as a thyroid receptor or vitamin receptor, because they receptor bind with hormone or vitamins, or they reside in the nucleus. So these receptors are composed of C terminal, N terminal, and the specificity of these receptors are this, that they are having a zinc finger domain or the DNA binding domain. So, as I told you that these receptors are reside inside the nucleus. So, for their activity, their ligand, for example, their ligand may be any enzyme or hormone. So, for the activity of these receptors, the ligand should have to transport across the plasma membrane, then cytoplasm, then should be enter in the nucleus where this ligand bind with the this zinc finger domain. So binding of the ligand with this zinc finger domain, what happened? This DNA binding domain of the receptor will be exposed, and then this receptor and hormone complex or receptor ligand complex that bind with this DNA. And after binding this receptor hormone complex with the DNA, then what happened? Then DNA synthesized messenger RNA, then messenger RNA translated into the protein, and this protein is involved in various cell biomolecular and processes and physiological responses. For example, cell division and cell proliferation, cell migration. So, this is how these receptors work. So, dear student, this is the book. You can consult for more um, reading from this book, and these are some web links. You can also consult this. This is one of the research article. Uh, you can also find the description of the different type of receptor from this research article. And thank you very much for your kind and attention. And please be safe from COVID-19. Take care. Allah Hafiz.